Good morning, everyone. I'm Gemma Molinos. I'm a reader in neuroscience and mental health at the IOPPN at King's College London. And it's always a pleasure to join the Scholars Day and see what everyone's been up to in the last year. It's also my pleasure to introduce Sam. Sam started his PhD in October 2020. So when the world was upside down and the studies were stopped, and he's always done excellent progress. And the same for the last year. He combines efficiently recruitment of patients with early psychosis from early intervention services with research. He has established great working relationships with all the clinical teams, and with any setbacks, he always has very positive attitude. So he's a pleasure to work with as a student and as a colleague. So now he'll take it on to give you an overview of what's happened in the last year. It's been very successful. He's got first paper published. He has another one in the pipeline. He's got a Warranters of Brain Travel Award, and he's got a Schizophrenia International Research Society Early Career Award. I'm just saying it just in case he won't, being modest about this. And so, yes, thank you for the opportunity to present. And Sam, over to you.
Thank you. Sam, yeah, would you share your screen and kick off?
Oh, there we go. I lost the control panel. Can you see my screen?
Yes, but you're not sharing the. Okay, now okay. we can see presenter view now, Sam.
It's because I got two screens going. Let me do that and just rotate through. How's that?
Perfect.
Okay. Well, thanks, Gemma, for that introduction. So yeah, I'm now entering the third year of my PhD. So I'm going to be talking a little bit about a sort of background to my main project and the literature review that came from that. That's sort of the introduction to my PhD. And then I'm going to talk about the main project, kind of give a little overview, a little update on where we're at with recruitment and that side of things. And then I'm going to finish off by talking about a little sub-project that I've been working on with some existing data while this data collection is ongoing. So just a brief overview of psychosis. We haven't talked about it yet this morning, but psychosis is this state that's a cluster of many symptoms sort of categories. So it's positive symptoms, hallucinations, delusions, disordered thought. But I just want to, even though that this is the focus of my PhD, I want to acknowledge as well that there's these other symptom categories as well: flattened affect, poor attention, memory impairment. And psychosis, the onset is usually in early adulthood, affects roughly 1% of the population, and there are really high both personal and societal costs associated with psychosis. And there's still a big problem of little progress with new treatments. So a lot of the treatments we do have for psychosis, although effective in many people, they aren't effective in others, and it really hasn't been any development of new treatments for quite a long time. So one way that I like to view psychosis is if we have these psychotic symptoms at this behavior level, one way that we want to try to understand what are the causes of these symptoms is to try to understand what's going on at these more kind of molecular levels. And one of the ways we can measure these is through different neuroimaging techniques. So these are some of the things that we might try to detect. You don't have to worry about what all these things are. I'm going to kind of guide you through an overview of what my project is investigating. But if we have these environmental stressors that are acting on people, things like bullying or social isolation, and these can strain brain systems associated with these responses, responding to these stimuli. And that can cause network dysfunction. It can also cause hyperactivity or hypoactivity in certain regions. And one region that we think is particularly important in responding to the sorts of stresses that occur in people with psychosis is the hippocampus. And hippocampus is involved in many different things, but one finding that we find quite often is hyperactivity in the hippocampus in people with psychosis, but also in people at risk for psychosis. And one reason why we think the hippocampus might be particularly impaired in psychosis is because it's a very finely tuned region with lots of inputs, and there can be an imbalance between this excitatory and inhibitory circuitry. So some neurons help to encourage neurons to fire, Others are sort of helping to stop certain neurons from firing to help guide collaboration in these larger networks. And an imbalance between the circuitry might cause a cascade of wider network dysfunction. And one possible reason for this might be what we call reactive oxygen species, oxidative stress. You've probably heard of free radicals and antioxidants. Well, anytime that your energy is using body, anytime your body is using energy, sorry, there are byproducts of this reaction. And these byproducts can cause damage to cells if not properly taken care of with antioxidants. And so a buildup of reactive oxygen species can damage cells, particularly those cells that use a high amount of energy, such as these inhibitory neurons, and that can cause a cascade of dysfunction throughout the system. And this was really the subject of my my review that I managed to publish last year, which sort of serves as an introduction to my PhD, where I talk about the developmental trajectory of the hippocampus in psychosis, the imbalances that we are aware of, and how those imbalances might be targeted with different treatments, different novel treatments. And this is relevant for my PhD because we're measuring a lot of these different things with different neuroimaging techniques. So a little update on that project. So there are 3 groups that I'm currently recruiting. I'm looking at healthy volunteers, people at clinical high risk for psychosis. These are people that have attenuated psychotic symptoms that don't yet meet clinical threshold, but they do have a very high risk of 20–30% of transitioning to psychosis in the next few years. And I'm as well recruiting people that are currently experiencing their first episode. So these are people that don't necessarily have people have been admitted to a service usually within the last 2 years, and they are still experiencing some level of positive psychotic symptoms. And we're measuring a few different things. We're measuring hippocampal hyperactivity through this arterial spin labeling of blood flow, regional cerebral blood flow changes in the hippocampus. And we're also measuring certain metabolite concentrations that we think are important. So GABA, this is a measure of these inhibitory cells I was talking about, and glutathione, which is the body's main antioxidant, and then therefore it's a sort of proxy for oxidative stress that I was talking about before as well. And so as you can see here, we're approaching the end of our recruitment. We still have a few people more to go, but we're hoping to all wrap this up by the end of September, by end of August. So while that data collection is ongoing, I'm currently as well working on another project that is using some existing data and sort of reanalyzing it with some new methods. This is a very long and complicated title, I realize, but essentially what I'm doing is I'm looking at blood flow data from these participants and analyzing it by combining it with some molecular data from PET imaging. So as you hopefully remember before, if you were thinking about these things across these different scales, if we can detect blood flow changes in people with psychosis, people at risk for psychosis, there's still a gap between our understanding of what's going on with these blood flow changes and what's happening at a more molecular level. Now, we can measure some of these molecular changes with techniques such as PET, but these techniques are really expensive. We can only image one of these systems at a time. So recently, a lot of these PET-derived atlases measuring different neurotransmitter systems have become freely available. And so we can combine these freely available atlases with some of this blood flow data to try to understand a bit more what's going on with blood flow changes in these patient groups. So I'm looking at 2 groups of already collected data, people at clinical high risk for psychosis gathered at King's, and a schizophrenia data set that was collected across a few American sites. And from these sites, I get individual regional cerebral blood flow maps of these individuals. And I then combine that with 19 PET atlases across a wide range of neurotransmitter systems. Now, you don't need to know what all these different abbreviated names are, but some of the systems that I'm measuring are dopamine, serotonin, glutamate, GABA, which you may be aware of. And there's actually 9 different neurotransmitter systems that I'm combining and that I'm looking at. And so how this works is I get these atlases, these 19 different atlases, and then I get a blood flow difference map between the patients and controls. So that gives us an indication of which regions are more altered in these patient groups. And so that's in schizophrenia and clinical high risk. And then I chop up I parcelate into 82 regions of interest using one of these atlases. And so then what happens is we get the average activity within each of those regions for each of these atlases and each of these patient groups. And so the simplest way to compare these things would be just to correlate them with each other. So then we could say, okay, well, in this region where there's high blood flow differences, is there high receptor density in these regions as well? But this gets a bit more complicated when we have 19 different PET atlases, and what is, some of these overlap, and so what is the unique contribution of each of these neurotransmitter systems? And so one way that I've tried to answer this is by using a computational method called the dominance analysis, and this gives us an idea by it's sort of a regression model that iteratively takes out and inputs these different maps in about 500,000 different combinations, and then it allows us to see what is the unique contribution of each of these maps. So again, there's lots of information here, but if we just focus on the top line, this is a schizophrenia group, and we can see that dopamine is important. So D1, D2 density reflected pretty highly, the highest of all these neurotransmitter systems, the blood flow differences that we see in schizophrenia. And that's perhaps not totally unsurprising. Antipsychotics act on the dopamine system, and a lot of these patients are taking antipsychotics. But I did as well control for antipsychotic use, and most of these effects are still here, but I'm happy to talk about that later. But we can see as well that there's some other neurotransmitter systems involved: serotonin, acetylcholine, GABA. And some of these systems are also involved in the clinical high risk, but we also see a little bit of a diverging pattern as well, where we see serotonin 1A and some other neurotransmitter systems like the mu-opioid receptors, norepinephrine, and NMDA. So sort of together, we see that some of the expected neurotransmitter systems are involved, but as well some ones that we think might be involved in excitatory inhibitory imbalance are different between the patient groups. So these are things like NMDA and GABA. So why are we doing this? Well, because we're trying to bridge a gap between these modalities and try to understand what's going on at a more molecular level, at this higher level of these blood flow changes. And this is important because PET studies are really expensive. An average PET scan might cost something around £5,000, and an average MRI scan only costs about £500. So if we can help to use these freely available tools to help create a cheaper biomarking tool, we might be able to detect these changes much cheaper. And then this is also a hypothesis generating kind of research. We're providing rationale for future people to go away and, okay, if we can see that NMDA or acetylcholine seem to be overlapping with the changes we see in psychosis, maybe we can repurpose some drug interventions to target those systems. So what's next for me is to write this up into a paper. I'm also going to be presenting it at SIRS in Toronto. This is a Schizophrenia International Research Society. And I'm finishing up the data collection for StressGap, the main PhD project. And yeah, so big thanks to the rest of my lab, to the co-authors on these projects, and to Mental Health Research UK.